Welcome to the Jack Westin MCAT podcast with your host, Ahmed Abdelkader.
And Eric Olson.
Welcome back, everyone, to another episode of the Jack Westin MCAT podcast with Ahmed and Eric. Today, we're going to be getting into amino acids and what they are, maybe how to group them as you study, and then all the amazing things that they connect to. And before we get started, Eric, anything new in the derm resident world?
Yeah, I mean, just getting ready to start derm residency. I'm halfway through my. Oh, I should say before. I keep meaning to say this at the beginning and not the end, but we are planning on doing our Ask Us Anything episode next week. So get your questions in. We'll take a look at them next week and hopefully get together with myself and Ahmed and Jaden, and we can touch base and help to answer some of the questions that you all have. As far as residency, yeah, I mean, just halfway through my last inpatient rotation, which is, if I was a pre-medical student, I certainly wouldn't know what that was. I might have explained this previously, but inpatient meaning like in the hospital, taking care of patients who are staying in the hospital, in the ICU, things like that. And outpatient being in the clinic. And so dermatology, of course, is a really outpatient specialty. I think I want to say in my 3 years of residency coming up, the next 3 years where I'll do exclusively dermatology, I think 2 months out of the 36 are like inpatient months where we do consults. So this is my last inpatient medicine, like being an internal medicine doctor this month, and I'm halfway through, and it's kind of funny. Like, it's obviously not what I see myself doing for a career, but as I get closer and closer to the end, it's a little bit bittersweet. Like, I think I've been thinking a lot more about kind of the privilege to be taking care of people and kind of the struggles that they have when they're hospitalized and trying to mitigate some of their suffering. So I think it's been good for me as a doctor to like think about that a little bit more and be reflective. So it's been an introspective time. Yeah.
I'm glad you're getting to experience that. And it's cool to see, your reflection and appreciation for the role that you've played in people's lives and your peers play in people's lives. And I'm sure, your patients are very appreciative of your efforts. So
how are your teaching sessions going? You got some students who are getting ready to take the MCAT. I'm sure people are taking it probably right around this time. Yeah.
Yeah. So it's awesome because I'm sort of in a couple of different areas, one of which being our live online course. And so I've got my cohort now that's wrapping up. Our last session is 2 weeks from tomorrow. And I guess people watching this have no idea when tomorrow is. The last session of my live online cohort is, I believe, May 29th. And so my students are getting ready to test, a lot of them in June, which is exciting because, it really sort of is the, insert your metaphor of choice, but the ribbon on top of the present of their med school application, which is exciting. Yeah. And then I've got some one-on-one tutoring students testing soon. A lot of them June 1st, May 24th. I've got one testing tomorrow, as a matter of fact. Had some get their scores back.
It's that time of year.
Yeah. I woke up to an email today, actually, from a student of mine who recently tested and scored a 522, which was unbelievable.
Wow. Incredible. Yeah.
Yeah. Just awesome stuff. And like I guess in a similar, but obviously very different to what you're experiencing inpatient, like you get this sense of appreciation and gratitude when your efforts translates into something tangible for someone else.
Into a 522, right?
Well, that specific student was, incredibly bright, and they started, high-ish, like,
Sure.
they weren't starting the 490s, but they made a pretty sizable jump. It's always great because, these are students that place their faith in you as an instructor and us as a company. And you really want to do everything for them to make sure that they're successful. And then when it pays off, it's awesome.
Yeah, that's
Um,
incredible. Man,
and it, yeah, sorry, go ahead.
I was just gonna say I'm thinking about the MCAT. I'm thinking that I'm just glancing at the date, and I took my MCAT on May 19th. Today for us when we're recording is the 15th, so it's like I don't think I'll ever forget the date.
Yeah.
It was such an important day. It must have been in 2018, I guess. An
Yeah.
exciting day. So anyway,
I actually couldn't tell you, I know the month I tested. I couldn't tell you the specific date though.
yeah,
But no, man, it's awesome. And especially when students like overcome, a challenge in a section that they've really been struggling with. Like I had another student also recently emailed me and was just overjoyed with her chem phys score. Like she had been stuck at a 125 for a while. And then we started working together in her last 2 weeks of prep. And on test day, she was at a 128, which is,
yeah,
those are the things that,
yeah.
little dopamine hit in the morning gets you ready for
That's
work.
awesome. Good way
But
to
in
start
any
your day
case,
with that email. Yeah.
oh man, it's awesome because and the last thing I'll say on this point is our students are obviously people too, right? And so you develop such a authentic or genuine connection with your students that transverses the student teacher relationship. It really turns into a lot of you almost become their fan, right? Like you're
Yeah.
rooting for them in a way that isn't natural of two strangers interacting, if that makes
Yeah.
sense. So
Yeah.
it's always great to see them do well. But in any case, in terms of doing well on the MCAT, I think one of the best ways to do that is to know your amino acids and the appropriate connections that are there to be made. And I remember when I was starting MCAT prep, I was like, there's no way I'm remembering all 20 of these things. It seems daunting. And then you factor in okay, I have to remember the amino acids plus 100 other stuff, right?
Yeah.
Or 100 other things. And where I want to start is how can we cluster them, right? Because for anyone with a psych soc background, you'll remember the term chunking, right? If you can group things together, it really improves your capacity for retention. And I think with amino acids specifically, there's a lot of information out there or suggestions as to different ways to chunk information. We'll go over sort of the ways that work for us in our prep, just to give students exposure to some of them. But obviously, you're welcome to do this however you like. And Eric, I'll start with you. Thinking back to your prep, do you remember, sort of how you went about studying the amino acids? Was there anything
Oh
that stood
yeah.
out?
I remember vividly because it was so terrible. Like you said, you see it for the first time, you see the chart, and you're like, this is like so outrageous that I would be expected to know all of this.
Right.
But it is. I mean, it's just one of those things you have to know. And it's not just like one of those things you have to know, but it's quite possibly the most important thing for you to know. If I were going to tell someone to memorize, because I think there's a lot of strategy on the MCAT and there's a lot of familiarity with the exam and how do they ask things and how do you make connections. But if there was one thing for someone to memorize, I would say it's the amino acids. I think, you could get asked a question on one of a million different topics, but you are guaranteed to get probably at least half a dozen questions that you need to know the amino acids for, I would say. You find it in every section. You can even see it in psych soc. I've seen it before. You have to know the amino acids. And so when I studied the amino acids, I had a chart and the chart broke the amino acids up into different subcategories. And I'd love to hear, Ahmed, kind of what categories you think are important. I think for me, you have the nonpolar amino acids. Then I would have amino acids with aromatic groups.
Yeah.
And those are kind of nonpolar, right? But I think
Yeah.
they're a little bit of a subgroup on their own. You
Right.
have polar amino acids. Then you have positively charged amino acids and negatively charged amino acids. And those are kind of the 5 groups that I would subdivide them into. And when I was studying, I would start early, to learn these because you're not gonna learn them in the last 3 days before the test. But
Right.
I would wake up in the morning, and I was in school while I was getting ready for the MCAT, so it was kind of, I had to have like my dedicated time. But I would wake up and I would write the whole chart out. And I wasn't just writing. I think you don't need to necessarily write, like, you can just do the R groups, which we'll get into the structure in a minute here, but
Right.
um, amino acids are all the same except for the R group. Like, it's the R group that's
Yeah.
different from one amino acid to another. So I would write out the R group. How many carbons in the carbon chain are we looking at? Is there a carboxylic acid? Is there a positive charge? Again, is there a hydrocarbon chain? What exactly does it look like? And then I
Yeah.
would write the name. I would write the whole name out: threonine, arginine, lysine. I would write the 3-letter code, and I would write the 1-letter code, and I would do as much as I could. And at first there were lots of gaps, and sometimes I was wrong on something or another.
Yeah.
And over time, I did that every day for probably 2 or 3 weeks. And by the end of the 2 or 3 weeks, then I knew them all. And for me, this is one of those areas that, like, personally, I don't really think flashcards are going to do it. It's different.
Yeah.
I think, to me, like, really writing it out, like, you are going to learn them. And this is not one of those things that you want to be second guessing or, like, not really certain of when you get to the test. Again, like, you are
Yeah.
going to have questions that require you to know these things. And so if you can look at an E and know that that's glutamate, that's the one
Yeah.
letter code for glutamate, then it's going to make your life that much easier and you'll save time. So for me, it was about drawing it out. It was about drawing it out. It was about looking at every component that I thought I needed to know. For those with PKAs, which I think we'll touch on briefly as well, like, I wrote down the
Yeah.
PKA. I wrote it all down. And in doing that so consistently, and after the 3 weeks, then I would write them down maybe once a week, once every 2 weeks, depending on how confident I was feeling. But after that time period of doing it so consistently, like, I knew
Yeah.
them, and I still, I wouldn't say I know them quite as well as I knew them 6 years ago, but I still know them, because, like, you really have to pound them into your brain.
Yeah.
But I mean, what was your strategy, and if you can tell us a little bit as well about kind of the structure of an amino acid and what they look like generally.
Yeah, and I mean, starting with the structure, it's an amino acid, and the name tells you a lot of what you need to know. There's an amino group, there's a carboxylic acid group, and there's a hydrogen, as well as the R group that Eric mentioned, all attached to this central alpha carbon. Right. And as Eric said, the R group is what introduces all the differences between the amino acids. And so it's a very simple structure when you think about it. There's 4 groups on a central carbon. 2 of them are in the name, right? And then you have a hydrogen and an R group as well. In terms of how I remembered them, I think it's important, an important disclaimer that I took the MCAT before taking biochem. So it was a little bit harder for me
Yeah, for sure.
to remember the amino acids. But I was like you in that I had to draw them out. Otherwise, I wasn't going to remember them. I tried to make sense out of things, right? So the categories that you mentioned there are very, very common. You got your nonpolar, your aromatic, your polar uncharged, your polar acidic / negative, and your polar basic / positive. That's really important to understand from a functional standpoint, right? Because that helps explain a lot of the different properties of the amino acids, and that's critical. But in terms of remembering the structures specifically, I sort of clustered them in specific ways, and I'll give you a couple of examples. For example, I clustered alanine, phenylalanine, and tyrosine together. Why? Well, alanine I knew started with an A, so I remember it was like very simple. It was just the 1 carbon. Phenylalanine is alanine with a phenyl group or a benzene ring attached to the carbon. And then tyrosine is phenylalanine with a hydroxyl. So it was a nice progression for me there that, okay, there's 3 amino acids and I get 1 from the next,
Yeah.
Awesome. And what's also cool about those 3 is that the 1-letter and the 3-letter abbreviations, relatively speaking, make sense. ALA, ALA, and then A, very straightforward for alanine. Phenylalanine is an F for the 1 letter because the proline takes P, but phenylalanine makes sense. There's an F there. And then it's PHE.
Sound, right?
Exactly. Yeah. And same with tyrosine, right? So tyrosine is a Y because threonine takes the T, but it's TYR, right? Yeah. And with the exception of 4 of the amino acids, I think all of the 3-letter abbreviations make a lot of sense.
Yeah.
So that was one cluster I had, and then I put the amides and the carboxylic acids together. So asparagine, glutamine, aspartate, glutamate, because again, alphabetically, it makes sense. A comes before G. A has 1 less carbon, right? So there's 2 carbons to the functional group in the asparagine and the aspartate, 3 to the glutamine and the glutamate. And they differ in just whether it's an amide or a carboxylic acid. So just by knowing asparagine, using those 2 simple rules, I get to aspartate, glutamate, glutamine as well. And then with the one sort of place where things got finicky for me was with the basic amino acids, because some of those structures, specifically arginine and histidine, are a little bit weird. What I would encourage students to understand is, like, you're almost certainly not going to get a question that says which of these is arginine and have like single atom differences between the 4 answer choices, right? So like, take advantage of the fact that this exam is recognition, right?
Yeah.
For multiple choice from a structure standpoint. And so the way I remember them, lysine, I remember, is like a precursor to gamma amino butyric acid, right? So it's a long carbon chain with a primary amine at the end. Okay, so that's my lysine. I remember there's multiple carbons, and I was never the type to know exactly there's this many carbons, right? But like, I didn't really care, because again, the MCAT wasn't going to test me that way.
Yeah, and it really doesn't.
Arginine, I remember, had a triangle of nitrogens at the end, and then histidine had the imidazole ring with it was a 5. It was a pentagon with positive charge. That's histidine.
Yeah.
And so I really try to simplify things that way. And then you remember some of the other ones. For example, glycine, hydrogen as the R group. So that's great. Valine looks like a V in the side chain.
Classic.
Leucine and isoleucine are isomers of one another. So you just got to move a carbon around, right? Cysteine makes disulfide bonds, so it's got to have a free SH group at the end, a thiol group. And then, to take it a step further, it's the only amino acid that's R, which means that thiol group needs to be coming off the carbon directly attached to the alpha carbon, right? So the beta carbon, you could call it. Proline is kinked because the side chain loops around and interacts with the amino group, right? Methionine is like cysteine, but there's a methyl group coming off of the sulfur, right? And then just with that alone, the only 2 I haven't talked about are serine and threonine. And again, alphabetically, you can sort of use that to help. S comes before T in the alphabet. There's 1 less carbon in serine than threonine. There's some 20 amino acids.
I love that. I love that. Like, first of all, I love that last part. Just like the simplicity of that rule. Like, S comes before T, there's 1 more carbon because it comes after in the alphabet, right? Like, I love, those are the ways that you remember these things, right? Because you're gonna have to have these little tricks. I'll be honest, like, probably because I spent hours learning the way that I learned it. I listened to you talk, and I'm like, I would not have grouped them like that per se. But what I love about it is that it shows that you really thought it through. Like, you thought about, how can I connect these? Like, what mnemonics can I use to remember these codes and things like that? And I think that's really what's important. Like, whatever makes you really process and like make connections and figure out how different amino acids relate to each other, that's what's going to be helpful, because that's how you're going to see a lot of these questions on a test. Is it might say, how does this, which one of these amino acids is closest to the one that you're seeing? Or something like that. One association that I did like that you made was asparagine and glutamine and aspartate and glutamate. And the reason I like that group is because, and you talked about the 3-letter codes, that most of them make sense. I think they're all pretty intuitive, but the 1-letter codes, like when you're in the non-polar groups and, you have like isoleucine is I and valine is V and glycine is G, and like, it's all pretty straightforward. Once you get into some of the others, it can get a little bit more dicey, but to me, like, asparagine, glutamine, aspartate, glutamate, like, those are the hardest ones to remember. And so I kind of like that you grouped them together, because you could remember, like, these are the
Yeah.
ones that don't make sense. And for me, similarly, like not with asparagine and glutamate so much, but with aspartate and glutamate, I would think like, okay, A comes aspartate, A comes before G in glutamate,
Yeah.
and they look very similar. And so you have D and E. Like I knew one was one, and so I had A is first, and so is D. Then
Yeah.
G is second in the alphabet, and so is E, right? And so like little things like that that can help you remember those codes
100%.
is going to be really
Yeah,
helpful. And you really do need to know those 1-letter codes.
definitely. More so than the 3-letter ones, I would say.
Yeah,
Because you
agreed.
see a lot of like
100%.
that notation where letter, number, letter, right? D105C, right?
Yeah,
I had to make sure
for
that was
mutations,
valid.
right?
But exactly, it's point mutations. That's really helpful. On that note, with those 4, like the amides and the acids, I sort of remembered it as the acid 3-letter code made sense. So ASP for aspartate, GLU for glutamate.
Mm-hmm.
The amides, because they have a nitrogen in the functional group, have an N in the 3-letter code. So it's ASN, GLN, right?
Exact, but that's perfect. I love it.
And
So go ahead.
yeah, go ahead.
I was just gonna say, as I listen to you talk, and like I don't listen to a ton of podcasts, but like I'll listen to them here and there. I certainly listened to them when I was getting ready for step 1 and step 2. Like sometimes when you listen to someone talk through a concept, especially something like this that's so visual, like you have the structures that you need to see in front of you, it can be a little bit difficult to follow. But I'm like not looking at the structures or the codes or anything in front of me, and I'm just I'm listening to you talk. I listened to your whole thing. You went through most, if not all, of the amino acids, I think, and I was able to kind of follow. And I was visualizing the structures, and I was looking kind of back in my mind at what the codes were and everything. And I think that's because I have some level of mastery of the amino acids. And so I guess what I would encourage our listeners to do is like maybe listen to that and you're like, what is going on? Like what are these? And like we can't explain them all to you over this podcast, right? Like at a certain point you need to go, you need to look at them, you need to draw them out. It's going to be very helpful for you. But I want y'all to get to a point where like you can come back to this episode and listen through what Ahmed said and be like, yes, I know exactly what he's talking about. I know what these look like. I know what these codes are. I know what the charges are. I know I can see these groups in my mind because I know these amino acids. And, I think that's the level of mastery that you're going for, is that you can kind of go through this sort of a discussion and really be able to follow along to all the nuances that we're talking about.
And, to your point there, Eric, like you said, like as far as the structures go and some of the memory tips and tricks, we'll say some of them are maybe atypical or not common, the ones that I use. And, I think there's a bigger picture there, which is like, come to these conclusions on your own, guys. Like,
exactly.
I think what makes, what makes this easy for me is a lot of those connections I came to on my own, right? Like, and there's value in, accepting or embracing, a way to conceptualize things from a book or from a course or from online. Like that's how you learn, right? But there's a ton of value in making those connections for yourself, that even if it means like taking something you see online or taking something you see through whatever medium and putting your own spin on it, or it can be as simple as, okay, I understand why this makes sense and I can recreate it, right? And be able to explain each step. I think that's huge. And, the other side of that coin is like, there's countless ways to learn any given topic, right? And so it's not about better or worse, it's about better or worse for you, right? Like what makes the most sense for you, stick with it, right? And somebody else groups the amino acids a different way because that works for them. If it's efficient and it's accurate, then it works for me too, and so that's important, and especially as we get into connection, right? Because with amino acids, there's a lot of things that it connects to. And, I might just run through a couple of these just to introduce them, and we'll dive into them in more detail. But like from amino acids, you can get into functional groups, right? Amides, carboxylic acid, that takes you down into OCAD. the synthesis of the synthesis of a protein, where the lone pair on the amide nitrogen, the primary amino group, excuse me, attacks the carbonyl carbon of the carboxylic acid, right? There's a nucleophilic attack there. You're into OChem yet again, right? Water molecule is released in that process, which means the enzyme that breaks down proteins is a hydrolase. That gets you into two areas. It gets you into dehydration synthesis reactions. Now you're looking at sugars and fats as well. And you also get into the categories of enzymes, right? Once you get into the categories of enzymes, you get into kinetics, right? And you get into pKa's like we briefly touched on earlier, right? That gets you into equilibrium, which gets you into solubility and gives free energy, right? And, from proteins, you get into the different types of proteins and primary, secondary, tertiary, and quaternary structure, right? And, that's a lot of connections there in just, a few seconds, right? And being able to have a concept map where you can grow outwards from it and see how different equations relate, and not to spend too much time on equations specifically, but if you memorize equations in isolation on this exam, you're never going to see the end of it. One, you've got to understand them, and two, you've got to understand them in context. How does this relate to other things? And with amino acids, I think, that's a great place to start because there's connections into so many different areas. And I'm curious, Eric, when you were studying, was there one or a handful of, those types of connections that really resonated with you that made things easier for you?
I don't know if I would say I don't know if I would say anything that made it easier for me. I think you make an excellent point, and I'm frankly impressed. I wish I was able to do what you just did in naming off all these areas that they could ask you about. And that's really what we should all be aspiring to as we prepare for the MCAT, is to have those concept maps. Because what they're going to do is they're going to give you a passage where they say, they might show a structure of a protein, and they might say that there was this substitution at such and such, at such and such amino acid position in the structure or whatever. It could just be mentioned in passing when they're talking about acidification of, muscle proteins or something. They could just mention amino acids in passing, and then they could ask you about any of those things that Ahmed just said. They will take the mention of an amino acid and run with it as far as they can. And so I think, when Jaden and I talked the other day about kind of how to look at passages and how to break them down. And one of the things that I like to do is when I look at passages is to really key in on concepts that I think are high yield concepts that I think are going to get asked about, things that are relevant, right. Things that I studied, and amino acids is top of that list. If it ever comes up, it should be alarm bells in your head, because it's just so high yield. It's just going to come up, and there's no way around it. You know me, Ahmed, I'm a molecular biology guy. So when I think about amino acids, I'm thinking about central dogma and protein construction and stuff like that, right?
And I didn't even get into that.
C, N terminal to C terminal. What's the start code is it AUG?
Yeah.
So you always start with a methionine, I think. That's something that could be tested. That could come up.
Yeah. Definitely the polarity of the protein, 100%. And then the way it's translated, and different mutations. And
exactly. I think looking at I want to just touch on a couple special cases if I can. Ahmed. One is glycine. I think probably everyone is aware. All these amino acids are chiral, which means that they have 4 different groups coming off of that alpha carbon.
Right.
And so there's a certain what is it, D or L conformation? Is that right?
Yeah. So,
am I
right? In eukaryotes, we use exclusively the L amino acid, and I just remember it's the opposite of the sugars, so that means we use D sugars.
There you go. So all of these amino acids are chiral. Apparently, we use L. I had forgotten that, if I'm being honest. But glycine
you don't use that in dermatology?
I don't use, no, not frequently at least.
Fair
But
enough.
the glycine has two hydrogens, right? Like every amino acid has one hydrogen coming off of it, off of the central carbon. And in glycine, the R group is just a lone hydrogen atom as well, and so that's a non-chiral structure. And so, again, just kind of a fact to know. Glycine can lead to like kinks in a chain of amino acids. It can be like a turning point or hairpin point, as well as proline, which has that hydrocarbon ring in it. Methionine, as we said, is like the first amino acid in any growing protein. And cysteine, as you said, creates disulfide bonds, which can be used.
Yeah.
You briefly mentioned protein structure and quaternary structures. And
Yeah.
that's where we see a lot of quaternary structures is with disulfide bonds of cysteine. And again, this is like not entirely exclusive to this discussion, but again, because I'm a molecular biology guy, I have to say one thing that we should probably talk about at some point is like laboratory techniques.
Yeah.
Western blotting. I worked in this lab. Maybe I've told this story before, so I apologize if I have. But I worked in this lab for a summer between years of undergrad, and I was working on a Western blot of this protein for whatever reason. And it took me like a month to do it right. Like I did it over and over again. I had done it with a supervisor a couple of times. I had done some other kind of practice runs and it had worked fine. And then she was like, okay, you can do this one on your own. And it took me a month and I kept doing things wrong, like over and over again. Western blot is used for quantification of protein, right?
Right.
And in preparation for running the protein on the gel, you treat it with beta-mercaptoethanol, which breaks the disulfide bonds. And so it kind of breaks down that quaternary structure so you don't have these like massive proteins. Anyway, it's one of the like great failures of my life is that it took me I used so much resources to make this Western blot work, and like finally it worked, and it was a moment of triumph for me. But it was still, a moment of shame because it had taken such a long time. And I tried to troubleshoot on my own. And eventually I had to go back to the supervisor and be like, I've done everything I can do and it's just not working out. And I was like misreading one of the measurements of protein incorrectly. And so long story short, like I was loading like 1,000 times as much protein as I needed to into the wells to be run on the gel. And so it was just like a mess because it was so much protein, like it couldn't even handle it.
Right.
And ultimately we sorted it out. So, but anyway, another like application sort of thing, they could jump into blots and start to talk about things like that. So just ways of like connecting amino acids to other subjects. I think they are not shy about, and conversely, like, maybe they're talking about an enzyme in the passage, and then they're going to ask you about amino acids. And one of the things that I think is important to note, and I would love to hear your thoughts on it, Ahmed, is like, I feel like personally, one of the question types that I remember seeing a lot of is that they'll give you, like, they'll show you a structure of some protein, they'll show you an active site, and then it'll be like, oh, there's like a glutamate right here, like in this active site. And then one of the questions will say, like, which of these other amino acids could you substitute for the glutamate, and it would change the efficacy of the enzyme the least. And so effectively, like, you need to figure out which enzymes are grouped together or which amino acids are grouped together. Just kind of like we're talking about, like, where's the structure similar? You have to be thinking about steric hindrance with some of these larger what's the word I'm looking for? Aromatic groups and things like that. But where else do you see like questions like that, or what kind of questions do you anticipate seeing that involve amino acids?
Yeah, another common one is like, it'll give you the ligand properties of a certain enzyme, and it'll ask you, what amino acids line the active site. Right? You've got, hydrophobic and anionic ligands. You've got hydrophobic and basic amino acids.
Because those basic amino acids are going to be positively charged, right?
Correct. Yeah.
Yeah.
Another very specific question that I saw once that I thought was an awesome question. Because, to your point there, Eric, you said, which amino acid substitution would have the least effect? And so in your head, you're thinking, which of these amino acids is most similar. And there's
Yeah.
one major exception to that I can think of, and it has to do with the phosphorylatable amino acids, right?
Mm.
So say the passage tells you, phosphorylating serine, 75 activates the enzyme. Which of the following substitutions would, most preserve enzyme activity, right? And then you look for a threonine or a tyrosine and you don't see it. And you're
Mm.
like, well, what the heck? I know I need a phosphorylatable amino acid. And the catch there is that when you phosphorylate an amino acid, you introduce a negative charge. And so replacing that serine with an aspartate or a glutamate basically keeps that enzyme constitutively active.
Yeah.
Right. And so that's a connection that I remember actually getting wrong on a practice test when I was studying. And I always share that with students because conceptually it makes sense, but it's just, it's those two wires maybe aren't connected when students see it for the first time. And so just making that connection in advance can really help.
Yeah.
But in terms of other types of questions, those are a lot of the structural ones. Which amino acids would be in the transmembrane region of a membrane receptor, right? And like I
Non-polar.
wouldn't memorize all these things. I would just understand it, right? Transmembrane,
Yeah.
trans meaning across the membrane, right? So the inside of the phospholipid bilayer is the fatty acid tails, right? So very non-polar. We know that like dissolves like, right? So we want non-polar residues in that region, right? And typically with those types of questions as well, you can really look for the odd one out. Like you'll have an alanine, a glycine, a valine, and a lysine, right? Lysine is the odd one out there. So that's
Yeah.
the one that you're
Yeah.
not going to expect, right? Now, I wouldn't do that blindly. I would use that sort of in tandem with, a factual understanding. But typically, and I don't want to say always, because it's definitely not always, but if you know your amino acids, a lot of the amino acid questions on the MCAT end up being freebies. Like, this structural change, like, what, or if you make this substitution, what will happen? Well, the hydrophobicity will go up and the steric hindrance will go down, something like that. Um, you know, it's a lot of like to the point questions. They're not big wordy questions usually with the amino acids. And so they can translate into a lot of free points on test day. Granted, some questions are definitely harder than others, but I would say, it's in terms of difficulty of the questions, amino acids wouldn't be at the top of my list, which is a good thing because of how high yield it is, right? Like if the
Yeah.
highest yield
Yeah.
topic also had the hardest questions, that kind of sucks,
You'd be in
right?
trouble.
But
Yeah.
I don't think, and again, everyone's entitled to their own opinion, but I don't think that's the case with amino acids. Like for me, off the top of my head, like, I had a lot more trouble with metabolism, different enzymes and different, regulations and things like that. And, there were other things that I found to be trickier, but
Yeah.
I think the moral of that story is like if we can understand, how the structures and properties translate, pun intended, into the functions of these bigger proteins, it makes it easier now to apply that in a number of different settings. And we touched on those connections earlier. I'd really encourage our listeners and viewers to go explore those connections now on their own. Because as we talked about when we were going over how to group the amino acids, right? The value in that comes from creating those groups and creating those connections on your own, right? And so I would encourage, like I said, our viewers and listeners to go through that process, and see what connections they can make, right? And then anytime you to a point where you get stuck, find a little hint or a breadcrumb online that you can use to keep going. And it bodes so well beyond amino acids as well. Just studying in general, right? Like, obviously someone is going to give you information in the form of a lecture when you get into med school, or there'll be a recording or a case-based study or whatever. But, the challenge now is like, how do you take that information and put your own sort of stamp on it, right? How can you retain it in a way that makes sense to you, right? Because you can phrase the same thing multiple ways. It's just finding a way that's going to stick and make sense for you.
Yeah.
And that's the biggest thing.
I do just want to add, I totally agree. And, I think we harp on this a lot on this podcast. It's like, we're trying to help, but you can't just listen to us. Like, this is the most passive
100%.
thing you can do. And you have to like put your mind to it. Just like Ahmed, like he created these groupings. And I was impressed by that. Like he really thought through, like, how can I make these make sense? Like, it's not someone feeding it to me. It's like me thinking it through on my own. This is what I think. And I also do just want to emphasize, like, I agree with you that you say the amino acid questions are the easiest ones, or some of the more doable ones at least. And I think what you said like, pick the odd one out. If you have like alanine and valine and glycine and lysine, then lysine's the odd one out. And like the way you were that fast about like, here are your options, this is the obvious outlier. You know what I mean? Like everyone should
Right.
have that degree of familiarity with it. I think that you can just pick it out, right? Because if you have to sit down and like write them all out, like you just don't know them well enough. And I think that these amino acid questions, for the most part, are going to be either you know it or you don't. And if you know it, you're going to know it just like that, and that's what makes them doable. And if you don't know it, then you're kind of in a bad spot. Again, not only because this is so high yield, but because you can't, unlike a lot of things on the MCAT where you can kind of reason your way out of it. Like you just have to know the amino acids. Like you have to know them. I'm telling you all right now, like you have to know them. If you don't know them, you need to learn them. If you have 2 weeks until the test and you don't know them, like that is what I would recommend. When I had students who didn't know them and they were getting ready to test, I was like, this is what you're going to do. You're going to take practice tests and review them, and you're going to learn the amino acids. Like those are the things. So I don't think we can overstate the significance of this for the test. We did want to talk briefly about some of the kind of like pathophysiology of disease, clinical applications. Ahmed, will you tell us a little bit about some of that and how it fits in with cystic fibrosis, if you will?
Yeah, and I mean it's wild to think about how such seemingly minor changes in a protein structure can cause these really serious health outcomes. And you mentioned cystic fibrosis there. Cystic fibrosis is the deletion, or it arises from the deletion of a single phenylalanine residue, I believe at position 508 of a chloride ion transporter in epithelial cells. And, just take the number 508, right? Like it's the phenylalanine isn't the last amino acid in that protein, but like several hundreds of amino acids large. Just getting rid of that one amino acid just causes some really, really bad outcomes, right? And so the removal of that one amino acid causes misfolding of that transporter, which really limits the amount of chloride that is pumped out of the cell, which limits the amount of water that then follows the chloride ion into the epithelial lining of the airways, which causes the mucus in that airways to get really, really thick instead of being more fluid and easily expelled. And that causes a whole host of issues for affected individuals with cystic fibrosis. And if I remember correctly, most people with cystic fibrosis die at a fairly young age. I believe under the age of 20, if I remember correctly.
So there's a relatively new therapeutic drug, and it's only, I think, in use for patients with so there are different mutations that can cause cystic fibrosis, and delta F508, delta being deletion, which I don't know if that's on the MCAT or not, but delta F508 is the most commonly seen mutation that causes cystic fibrosis. It's autosomal recessive, excuse me, so you have to have 2 copies to have the phenotypic cystic fibrosis. But there's a new therapeutic drug, and I'm trying to remember the name. I think I know what it's called, but I'm not sure, so I'm not going to say because I don't want to be wrong. but it's only for use in patients with the Delta F508 gene mutation, and it actually is extremely effective. I remember I was in a pulmonology clinic during medical school, and we had some patients come in who had this disease, and like really turn their life around, being able to take this disease. Some who had become like endurance athletes,
Oh,
and
wow.
like were really, I think prior to this drug being available, a lot of patients did have kind of premature death. I don't know if I would say under 20, but certainly, in their 30s and 40s. Um, but some people like took really good care of themselves and like sometimes got lucky, or whatever the case may have been, and survived longer than that. But this has been a game changer drug for these patients who have that specific mutation. And it's also interesting. I don't know off the top of my head the mechanism of action of that drug, but it is interesting because there are other mutations within the cystic fibrosis protein,
Right.
or whatever that transmembrane protein is called, that cause cystic fibrosis, and you develop the same phenotype, the same disease, but because of the way the drug interacts with the protein, like maybe a different mutation is does not allow the drug to work as well. So it is
Huh.
interesting, like one mutation versus another can have specific effects for how we respond to, pharmacologic intervention and things like that. But yes, in answer to your question, it is, a very severe disease. And patients
Yeah.
will get, they get really bad pneumonias because they aren't able to clear the mucus. It affects the
Right.
viscosity of the mucus in their lungs, and so they can harbor bacteria. And they get these pneumonias with like kind of atypical, I shouldn't say atypical, but like kind of out of the ordinary bacteria that don't cause pneumonias in like healthy people. And then
Right.
they can also get like pancreatic insufficiency because the pancreatic fluid that's supposed to flow out of the pancreas into the bowels gets really thick, similarly to the mucus in the lungs, and so it isn't able to flow out as effectively. And so a lot of times with those patients, they need to take supplementary pancreatic enzymes because they just don't have pancreatic enzymes flowing into their into their bowels. Excuse me. So
Right.
it is an interesting disease, but yeah, it's incredible how like one deletion in a single amino acid can cause such a significant phenotype. And go ahead. Sorry.
No, I was just gonna say it's also incredible to see how research in medicine can drive some really incredible developments, like I don't want to say completely, but to largely mitigate cystic fibrosis through the
Yeah.
mechanism of an oral pill. I'm assuming is you know really impressive and really significant, right? Obviously, in the lives of those people, as you mentioned.
Yeah, a lot of our listeners will go on to do clinical medicine and, treat patients in the clinics and in the hospitals and whatnot. But I'm sure some of them will go on to become researchers, and, some of that may be in clinical medicine, and some of it may be in basic science and drug development. And, this is like foundational to those sorts of things. You know, many of that work is done by PhDs, right? But there are certainly roles for physicians, and some of our listeners may go on to get PhDs or other advanced research degrees as well. And when you do clinical trials, then you have physicians on board, and so there's certainly a role for us in drug development and things like that. The other disease we wanted to touch on briefly is sickle cell anemia. So cystic fibrosis being a deletion, right? Like we get rid of an entire amino acid. Um, and we could get into like, it's a deletion in the DNA, right? Like it's not a deletion in the protein itself. So you have the
Right.
deletion of three nucleic acids in the DNA code that causes the deletion in the protein of one
Right.
amino acid. But that's a conversation for another day. But sickle cell is not a deletion; it's a substitution, and it's a substitution of a glutamate amino acid for a valine. And so, thinking about like what do these amino acids look like? Like a glutamate has a carboxylic acid functional group that's negatively charged and polar, right? Whereas a valine is kind of a small hydrocarbon side chain, which is nonpolar. I don't know what exactly that does, but it changes the conformation of the beta chain in hemoglobin, right? And so when you have, periods of stress or hypoxia, patients aren't getting enough oxygen, and they can have these sickling events where, hemoglobin in red blood cells are supposed to look like, little saucers where they carry oxygen more or less to different parts of the body. And when you have this, often triggered by hypoxia, this sickling, as I'm sure probably most of us are familiar, then the red blood cell will change shape into this sickle shape. And again, like structure of the protein affects function, and the amino acid chain and the amino acid composition is what's making up the structure of a protein. And so the structure of the beta chain kind of gets out of whack, and you get this sickling, and it causes missaping of the red blood cells. And so they can kind of stack up on each other. They get stickier. They can cause, clots. They can cause pulmonary embolisms. They can cause kind of micro infarcts. A lot of times these patients will have clots in their spleen, or, in the vasculature leading to the spleen, and they'll have what we call autosplenectomy, which is like they get this like hypoxia and ischemia of the spleen where they're not getting enough blood supply and they're not getting enough oxygen flow. And the spleen will kind of just die by itself over time. And so, they're susceptible to different sorts of infections that our spleen helps us to keep at bay. They can have these pain crises when they're not getting enough oxygen, often have bone pain and things like that. So it can be, it's also a very high burden of disease, or I shouldn't say burden of disease, but significant symptomatology with these patients. And again, like you talked about, the cystic fibrosis protein being like hundreds of amino acids long, and I don't know how many amino acids it is either, but at least 508, right?
Yeah.
And you're just losing one amino acid, and it has this phenotype of like people are getting pneumonia and people's pancreases aren't working. Similarly, like this is not a deletion, it's a substitution. So you still have an amino acid there, but again, it's no longer negatively charged. It's nonpolar and hydrophobic, right? And so whatever that one amino acid is doing makes such a significant difference. And so, just understanding like the implications for this in disease, and even moving forward into medical school and in your practice, like these are things that matter. These are things that matter to patients. We have therapeutics now that target certain ones of these, as we discussed with cystic fibrosis. And so it's very relevant to medicine today. Like we get more and more into the basic science as we understand it better, and we can use therapeutics to target those specific mechanisms of disease.
Right. And, we'll wrap up there. I think, understanding the real world significance of MCAT testable topics is a really good way to keep learning exciting. And also to, in some cases, begin to envision what a career as a physician could look like. Obviously not everything on the MCAT is directly related to being a physician, right? Like I don't know many, physicians that worry about beta and gamma decay much, but, there are things, there are things like these with amino acid substitutions that, manifest into, some serious diseases that, us as future physicians may be responsible for treating, or, as researchers, trying to cure. And so having that perspective is always helpful. But anything you want to add there, Eric?
No, just, draw it out, quiz yourself, learn the amino acids. You have to do it. There's no getting around it. And it's possible, as we can attest.
Yeah, definitely possible, definitely doable. That spaced repetition, though, is really helpful, right? Continue to reinforce that information over time. And, take advantage now of your strength in that topic, make those connections as we talked about, and capitalize on those freebies on test day, because, having questions that you can look at and say, yep, like I'm certain I got that right. It's just a big confidence boost come test day. And, that confidence element is really underrated, part of scoring well as well. But in any case, thank you all for listening and or watching to this episode of the Jack Westin MCAT podcast. Reminder again to get questions submitted through the Google form that we'll have in the description below. And we'll, we'll be looking forward to answering your questions in our next episode, which will be the Ask Me Anything podcast, hopefully starring or featuring myself, Eric, and Jaden as well. And happy studying, guys. We'll see you next week on the Ask Me Anything episode. And as I mentioned, looking forward to answering everyone's questions. Have a good week ahead, everybody.